Hi, everybody. So I am Vanessa Hobbs, and this vendor session, I will be going over the biotechnology basics module and then the two different versions of that module that I have available. So I am Vanessa Hobbs. I have my master's in biology from Vanderbilt University and a second master's in chemistry from Middle Tennessee State. I have been a college professor since 2008, and while my intent was to go into forensics, I ended up becoming a part-time professor mainly because my husband was in the military and we moved a lot. And then having young kids, science is not really conducive to full-time work. And so it really worked with child rearing. So I'm currently an instructor at Johns Hopkins where I teach cell culture and stem cell culture and biodefense. And then we have a small lab in our home where my husband and I make proteins for a pie in the sky idea in the future. So how we started, I started out as the Hobbs Science tutorial in 2021 when I noticed that many of the homeschoolers did not have access to biotechnology techniques. So I started teaching protein purification at my dining room table, and then that quickly increased to biotechnology intensives and forensic classes. And really, I've dealt with students all the way down to 10. And we outgrew the dining room table, and now we serve students in Frederick, Maryland at the Frederick Innovative Technology Center. Overall, the homeschool science curriculum world is missing biotechnology options. So, as a homeschool mom, I know every single secular science option that is out there, and we do not have access to step by step, week by week biotechnology labs available. So, after a years of development, honestly, and equipment research, we do now currently have an option with some affordable lab equipment that recently has become available. So, what if we could take the techniques that we actually use in the lab and give them to your student to do? So, for example, your student would be using a pipette and loading an electrophoresis gel. They would be learning about the molecular methods and what's happening in their tube before they actually do it using hands-on methods. And then being able to run these techniques, I use these every day in the lab, and being able to see what we see. So we'll start out with dye, but then we'll move over to DNA patterns. Being able to know what these DNA patterns mean. Do they have high cholesterol? Do they have sickle cell anemia? And then videos from me where I'm showing you the gene. I'm showing you what that pattern looks like. I'm showing you where that cut occurs in the gene and why that ends up making two bands versus one and what happens when there's three. And so there's lots and lots of instruction that comes with this biotechnology. So even if you aren't the best at science, the videos and the hands-on help are going to be helping your student through that. So for the Biotechnology Basics Module 1, this does come in a kit form. So we utilize mini PCR equipment because they are the most affordable ones on the market currently. And so it wasn't until we got this transilluminator, which is what actually lights up our DNA, that we were able to actually do this and be under like $600 of equipment. But we use many PCR equipment that's shown here, and using these Edvotech strips, we can send out those reagents. It includes a 171-page color lab manual, which I'll show you a little bit deeper in that in a minute. And so that currently runs at $450 plus shipping. I am offering 10% off if you order it within the next week using the code CSEA. And so I am offering 10% off there. If you have issues where maybe you have an ESA that's not funded and it might not get funded till mid-September, please let me know and we can work something out where I can get that applied to any orders after that because I know not everybody has their ESA funded currently. This will include 15 labs focusing on the role of DNA and biotechnology. This is going to be great for an advanced science education student, middle through high school, um, but it could also be used in the place of your science curriculum. So this would be one full semester of courses of a course. Uh, and then also, oftentimes you need three sciences and an elective. This could also be as an elective. My focus when I run courses is hands-on. I don't want to be talking for 3 hours while you listen to me. I want a pipette in your hand. I want you running it. And so the gist of this lab-based for course is for you to be able to run the labs and learn the concepts behind them while doing hands-on interesting stuff. So this is the table of contents. In our lab manual, you'll notice that there are 15 different labs that we do. I do have safety notes that have been added in there. They are going to be learning about gel electrophoresis, how we set up a gel electrophoresis rig, how do currents work, what is DNA. They'll be determining whether somebody has sickle cell anemia. Does somebody have high cholesterol? Do they have the cancer gene? Is this a genetically modified organism? And then we end in lab 15, where you have to take a look at the genotype of two lion cubs and figure out which wildlife preserve they should be put in based on their genetics. So really, it's just taking these concepts of science that used to be on paper or in video and putting them in students' hands to be able to see this is the current use of science in the field right now. So kit components, what's as important is what doesn't come with it. So I send you all of the equipment as well as the consumables and the strips. And this is enough for one student to be able to run them. If you have multiple students, I do order refill kits so that you can do those and not need to order a new kit. Not everybody needs their own rig. I recommend sharing those, and they can also be sold and passed along to other people to use this curriculum as well. I don't include glassware, mainly just because I don't want it to break. Graduated cylinder. I do recommend a glass reagent bottle for your TBE buffer to hold that, but plastic will work fine if it's cheaper. Wax paper. Um, you can heat up our agarose gel in a microwave. I also will be sending out a plug for a Erlenmeyer flask to make sure the chemicals don't get out. Most of the chemicals are biodegradable and non-toxic, but just to be sure. And so I do send out, it's a Drosophila plug, but we use them to plug up the flask. So you can use your own microwave if you want, but most of us in science have a separate science microwave for about 10 obvious reasons. So when you receive your kit, it's going to look like this. Yours will come in two shipments, mainly because those reagents I need to get out and are temperature sensitive, and so those will likely come first. So you'll receive a reagents package and then this box. Inside that box, you are going to have a lab manual. And so that is the color lab manual. That looks like this. I have a prettier version, shinier with the logo on the back. And so yours will look slightly different after I got them back from the printer, but these are completely full color. I'm showing you the gene. I'm showing you where it gets cut. I'm showing you what those banding patterns look like through this lab manual. The reagents kit is what you're going to receive. It's going to come looking like this. It's going to include the gel tablets. Those are going to be what you use to make your agarose gel. It's going to include the TBE buffer powder, which all you have to do is add water. And then these DNA strips are going to be what we use to be able to put those very small volumes into the gel when we load them, which is what that video that you saw earlier. It will also include the Bandit kit, the which is the gel electrophoresis rig. This is the View It. It helps us to be able to see the DNA. And if you saw me at the conference, I had this run so you could see the glowing DNA bands. You get a box of pipette tips. And then the other really important piece of equipment that we have here is going to be our pipette. And so that you're going to be using through any of the future modules as well. So you don't have to repeat this by. And so this is the pipette that we use and that your student will be learning how to use. And it's a very common piece of equipment that we use in the lab. Now, as a homeschool mom, I definitely want to be taking a look at that lab manual. And so I do have a short video here that will show you pieces of that. If you go to my website, you will be able to find the first lab, and all of the pages before it. So I guess all the way through pages 32 or I guess 22. So you can download those and take a look at them. So I have that on my website. It will have the kit components. And then you'll notice that each of these labs have a QR code attached to them, and that's where the instruction is going to come in. So when you use those QR codes, it is going to take you directly to a video where I will be walking them through the basics of each of those labs. And so each lab has its own instructions. Some of these are like 15 minutes, other ones can run like 35 to 40 minutes. But of enough instruction that as a parent who doesn't have an understanding of science is able to do that. So this was our genetically modified lab. And so that's in lab 12. So they'll be testing to see if the plants they have are genetically modified. And then I go through this, I show you what the DNA will look like, and I show you how this works, and I show you why that banding pattern is so important. And so you are not left alone to your own devices on this one. I'm walking you through all of the steps. And in fact, in these videos, I'm loading them with you and I'm running them. And then at the very end, I also have a video of the gel run. So if you have a mistake or something doesn't go right, you have a copy of the results. And so I have full color, lots of pictures. They're going to be learning how to do comparison of genetic relationships, being able to determine whether the impact that those genes have on cancer, being able to determine uh, where you can send those lion cubs to the best genetic match. All of these have lab sheets at the end of each one that allow you to be able to record your results and be able to have a high understanding of all of the material that we teach. Now, I do know that not everybody has as large of a budget for the science. So because of that, I have turned this into an online course. So this is an online version through Teachable. And with that one, it does come with an instant download if you purchase it through Teachable. But if you want the 10% off, I'll have to do the main website and then send you over there. Um, but this one has everything that's in the lab manual without having to buy any equipment. So that one is much more affordable. You do go over the 15 labs. It has all videos, and the videos have been redone to where you are watching me load them and run them. And then in the lab manual, it'll have a picture of the results for you to analyze. Those are printable. And so that is asynchronous and fully online, and you can complete that at whichever pace that you would like. So I do have a more affordable option. We accept ESAs from the following states. Now, Florida is tricky. I have applied to be on their marketplace, have not been approved yet, but I do know that I am UA and PEP reimbursable. I actually am an approved tutor through the state of Florida, so that's another way I could go around it with some individual lessons if you needed those. And so that's kind of how I've figured out Florida by now. I'm still working on other states. Alabama and South Carolina do not do boxes, so I need to go back and reapply now that I have the course. So I may be able to get the course through them eventually, but we're working on increasing this list regularly. So what is coming up in the future? This is only one semester. Most people are going to want 2 semesters. So because of that, Module 2 is going to be coming soon. I hope to have that done by December. I'm also going to develop a lower level curriculum. So this curriculum uses DNA. And so for the upper level, I want to do just dyes. Um, and so that's really important to be able to get them access to that. So I am developing that. Biotech mini course, that one is going to be $20 so that you can give biotechnology a try. Real fun things you can do with pineapple juice and Jello, so things that you can do around the house and affordably. I'm also developing a biotechnology lecture course, so not the lab. If you don't want to do the lab, you can at least learn and discuss the specific techniques that we use. Now, I know that dropping $450 on a biotech curriculum when you've never heard the word biotechnology and don't know what it is a big ask. So I've been working with Mini PCR, which is the company that makes our gel rig and transilluminator. And they do have a free lab available. And so this is a PCR code or a QR code that you can use to request a free lab. So I have recorded a lecture, and then I will give you an access code for access to a free lab that you can do online. So I do have a limited amount of access codes, so I have to know how many I've given out so that everyone can get on it. They do not have the capability for me to give it out for everyone to get on yet. So that's the only way that the company and I have been able to work it out, where I can get people access to these labs. So this is a micro punter where your student will learn all about the International Space Station and figure out which part of the space station has been contaminated with bad bacteria, and then figure out which part of the space station needs to be clean. They'll go through gel electrophoresis, PCR. So it's really a great way to give your child a free, fun thing to do and figure out if biotech is something that they'd really be interested in. And so if you will give me your email there, I can then end up sending you that to your email. And that's instant access once you end up getting the form. This QR code is also going to end up linking to my website so that you can see all of the options that we have available that I'm hoping to expand in the future. And so we are hoping to work with people with ESAs. I'm just the general overall gist of my work is being able to increase the accessibility to biotechnology because that's something that many people don't have access to due to cost. And this is really our way of making that accessible to everyone.
Now, I went ahead and put the link to Serious About Science website, as well as the link to that form people can fill out if they are interested in that biotech at home lab opportunity. That's on the video as well as over in the CMate group. And of course, it'll be in today's daily wrap up email. Someone's asking, could you share that coupon code information again?
The coupon code, yes. So for 10% off all orders, including reagent kits, all of that, that is SEAC. And I'll put that through to set like for 1 week. And then, like I said, if there's an issue, because I know people are still getting their ESAs funded through September, I'm more than happy to work with you.
All right. So I will again put that in today's daily wrap up email as well for anybody who would like to check that out. We really appreciate the coupon code. It's always good to save a little money. We do have
Yes.
some questions coming in. You mentioned that not every you don't need a kit for every learner. They say that they have 3 kids from middle school to high school. Would one kit work for their whole family?
Yes. So you have an option of doing one kit where they practice loading. So, for example, every lab they'll load anywhere from 4 to 6 wells, and you could have like them load 2 wells, two, and then two. But if you have for 2 students, I can see sharing. Once you get to three, especially if you have more of a high schooler, I would get them their own reagent kit so they could run it on their own. But you do not need 3 bandits. You just need one main kit. And then module 2 is going to utilize the main kit. So I'm not going to make you buy that again. I'm trying to use as much of this equipment again and again.
So they would just buy the curriculum
Yeah,
and any add-on kit, but not the
correct. Yes.
not the actual technology itself.
Right. Yeah. The most important part in biotechnology, as anyone will tell you, is going to be the equipment. That is the most expensive part, and that is why it's not accessible generally.
You mentioned that that could be resold to other people using the curriculum. Do you have any way for people to connect to sell those used?
I would like to set up a page where people could do that. I don't have the capability. I mean, honestly, I've thought of everything trying to buy it back and sell refurbished. I just don't think I have the space to implement it. But I do, you would be able to sell those anywhere that you sell curriculum. I do have a way that you can purchase lab manuals extra as well. Even for the online one, I have a way that you can purchase the online manual. to be sent because I know some people just want paper, and I'm with you on that.
I'm gonna say I know that this is fairly new, and so there aren't a lot of people who have used this and are selling theirs. But I just want to remind everyone we do have a buy sell trade thread pinned in the featured post in the C group. So if you use this curriculum and you've completed it and you want to sell yours to another C family, that is somewhere where you could always list it for sale.
Yes, absolutely.
Um, there aren't I do want to be clear, there aren't any there now
No.
because this is new, but that is one place, and hopefully as Serious About Science grows, you'll have a Facebook group or some
Yes.
other way where parents can connect because that stuff is expensive.
It is, yes, and it needs to be reused. And I've taught at enough colleges that I'm so tired of it. I'm just like, please reuse these. Please use the refill kit. So like there is no reason to be wasteful on these resources at all.
Someone says you mentioned having a separate microwave for science or heating something from the kit up in your own microwave. If you don't have a second microwave and you don't want to use your family microwave, is there an alternative heating
Yes.
method?
A $20 hot plate on Amazon. Yeah,
Okay.
just buy a hot plate on Amazon. Yeah. There's a few ways to go about it. I do have the plugs that's what they use, what the company does when they don't have access is those Drosophila plugs. So I do include those in the kits just in case. And yeah, so there's a few different ways to get
There
around that.
are alternatives to contaminating your microwave.
Yes. Now, this is much safer than the chemicals I use to run gels. So these are non-toxic and biodegradable chemicals. So short of eating and drinking things, you're going to be fine.
We did have a couple of questions about that. If
Yes.
your curriculum has any kind of unit at the beginning that goes over safety practices and things like that for labs, and if there are instructions for disposal.
Yes, so I do have a safety section about the instructions for the disposal. So for example, the TBE buffer, you're only making 100 milliliters of that, and that one can be poured down the sink. Our gels are non-toxic and biodegradable, so those will go in the trash and they'll form like a thin film over time. If you leave them out long enough, you'll see what I mean. Um, and so we do have these are all BSL-1 low level labs. In the next module, we are going to be dealing with bacteria. So with that one, we do get into sterile technique, making sure our place is clean. And then obviously I wear gloves in the videos when I'm working with it, and that's something that you'll buy on your own.
Had a slightly scary experience, not actually dangerous by any means, but one of my kids did the petri dishes where you wash your hands versus hand sanitizer. And we had a hurricane last
Oh
October, and we
no.
our home flooded and we lost everything. But when we were going to, my husband was packing things up, I did not realize that he tossed those into a shoebox and packed them into something. And then 4 months later, I was unpacking some things that he had managed to salvage from the house, and I found those petri dishes.
Oh. Well, I'm not sure how moist they were because if you leave them, generally they just dry out into like a film.
Well, you could tell there was some moisture in there because there was some
Yeah.
mold growth.
Yeah.
So it was a whole nother experiment that had happened.
That's when you love the joy of bleach and you just bleach everything.
Yes, exactly. But
Yes.
I was like, what? Why? But he's not a science guy, so he had no idea whether or not this was something that was important and needed to be
Yeah.
saved, but also did not mention it to me in the chaos of hurricane
Yes.
cleanup. So it was just a surprise 4 months after we.
Yes.
Oh, the joys of homeschooling.
Yes.
Um, Another person wanted to know if they were going to get together with a co-op group of kids, would they be able to purchase the machines at a bulk discount if they wanted to get several, if this was something that they
Yes.
wanted to run semester after semester? And would they
Yes.
contact you or the company directly for
Well, I am the company directly.
that? All right.
So, yes, and those would be, you could get that into groups of four, uh, where you would do one kit and then just in the next year's doing ordering reagent refill kits
Hmm.
for each one. And then also I would be open to giving you the PDF copies so that you wouldn't have to worry about like Because honestly, printing anything nowadays, as you are familiar with, is insanely expensive. And so these do get a little more expensive than what I ever would have thought they would be. And so I would be willing to give PDFs to those schools so they could print off on demand.
Um, you mentioned an elementary curriculum. Would that be a full year course or would that also be a semester? And would it require the same technology and equipment?
So with that one, I generally start at a semester because I do them in chunks. And I guess since I've been a professor so long, I just do everything in semesters. It's the only, I get overwhelmed when you do a whole year. I got to start with like a semester or else I get overwhelmed. And so yeah, we'll start there and I'll extend it out with the elementary one. They're doing dyes, so they wouldn't need the transilluminator. And so that one would be cheaper because it's using dyes and you're actually not using real DNA, so that makes the chemicals a little bit cheaper.
Okay, so they would not need the same equipment then.
Not as much, no. They would need less equipment.
You mentioned that you don't send glassware. Is basic glassware like a glass measuring cup from the dollar store going to cut it, or do I need to order specialized chemistry glassware?
I would prefer an Erlenmeyer flask when you are doing the heating because that just makes it less prone to injury because of the shape of it. Because it's boiling and then when you it just works better. So I highly recommend that. I have used beakers in the past and cups. I'm not there to stare and judge in any way. So I always when we heat things, we always try to do Erlenmeyer's just to minimize the solution loss when we heat things up. So it's just a science nerd thing in me. So there are ways you can get around it.
Do you have any recommendations for chemistry glassware that is affordable but well made for someone putting together a home science lab?
Honestly, the amount of things you can get on Amazon is disturbing. And you just buy whatever is on there. I mean, our I buy normally the cheapest glassware. The only issue I have is when I try to sterilize things, sometimes my glass cracks because it's not high quality stuff. So I always there's plenty of very, very cheap overseas science equipment available on Amazon that I utilize on a regular daily basis.
We are about at the end of our time. I want to thank you again for coming on, especially when you're not feeling well and sharing all of this information with us. I've got the discount code and the links to your website and the link to that free lab offer. I want to let everyone know at Vanessa's session in June, there were a lot of questions about how to do a strawberry DNA lab that she mentioned. So if you have not looked in our summer magazine, which is linked in the conference emails, there is that at home lab instructions that she wrote up, as well as a wonderful, wonderful science article that I highly encourage everyone to go check out. And Vanessa, I hope you and I can brainstorm when you're feeling better about what you
Yes.
might want to write for the October magazine
Yes,
as
I hope
well.
so. And I hope to be back for more talks when I'm not sick.
Don't be sick in November, okay?
Yes, that's when I always get sick again.
No, no. Get sick in October
I
this
will
year
work on it.
or
I
December.
will.
Schedule your illness
I'm
better.
gonna need that petri dish from earlier.
Well, go get some rest and enjoy the rest of your Sunday best you
Thank
can.
you.
Bye.